Okay, so you're happy, Mark? You're good? All right.
Yes, all,
Okay.
all good, thank you.
Let's do an introduction. Okay, so hi everybody. It's delightful to not quite see you through the Zoom link here. Welcome to another Genomics Aotearoa seminar. And today has been a fantastic day so far. The biochemistry building in which we are host has flooded, and so we're all swimming. And we've all gone home in the hope that we can dry out. And which is great because then I get the opportunity to focus more strongly on this particular talk. So today we have a really special seminar from Dr. Mark Eldridge, who is a senior principal research scientist at the Australian Museum. And he's going to be talking today on conservation genomics and performance management of the warru in arid South Australia. So technologies that are really close to our heart and things that we're also trying to do with some of our species. So it was great to see how things are doing over the ditch. So Mark, if you're all good to go,
Yep,
I'll
all
turn
good.
it on to you. I will turn off my video and it's in your hands.
Great, thank you very much, Peter. And yeah, it's a great privilege to be talking to you guys this afternoon. Given the sort of week the world has had, I figured that something slightly uplifting and positive might be a good thing this afternoon, something not too heavy. So this is really a case study of one particular species management in South Australia and how we've used genomics to inform the management of that particular taxon. So it's kind of fairly heavy on context and background, but I hope it will still be useful for you people working in similar fields across the other side of the Tasman. Firstly, I just want to acknowledge that I'm speaking from the land of the Gadigal people, which is where the museum is based in Sydney, but this work was done on the Anangu, Pitjantjatjara, Yankuljatjara lands in South Australia. And as it turns out, lots of really close links with those people, some of which I'll talk about in the rest of this seminar. What I wanted to do, first of all, is just give a bit of background about how I ended up where I am. And so for me, I grew up in this part of the world, in the far north coast of New South Wales, in the Tweed Valley, which is a really lush coastal volcanic valley. One of those rare parts of Australia which actually looks a little bit like New Zealand, I have to say. But up there in this town called Murwillumbah, it's very much dominated by, Tweed River and by that big mountain up there in the background, Wollumbin or Mount Warning. And so I grew up in this really biodiverse area as a biodiversity-obsessed kid. Didn't really know what to do with myself as a career, but then ultimately went off to uni to study biology at Macquarie Uni in Sydney. So I migrated slightly northwards. And there I discovered what turned out to be two great loves and passions. First, I discovered the power of DNA and genetics and genomics in terms of examining biodiversity. And I also discovered rock wallabies and marsupials, Australian marsupials, and that, there's a group here of animals that we knew very little about, but which genetics and genomics would be incredibly powerful and help us to understand not just their evolution and taxonomy, but also contribute to their conservation. I then moved from Macquarie Uni to the Australian Museum, also in Sydney, where, it's not just public displays of stuffed animals, but there's also behind the scenes with the collections and a fully equipped DNA lab and tissue collection of frozen tissues. So it's a really great place to work in terms of being able to do genetics and genomics. So the rock wallabies themselves are this really biodiverse genus spread all through the rocky areas of Australia. There's 17 species, and they've managed to colonise basically every patch of rocky habitat in this whole continent. They're quite recently evolved group, still currently actively speciating in many ways. Widely distributed, as you can see, in the ranges, and about half of them are listed as threatened, either vulnerable, endangered, or critically endangered. main threats are the usual threats in an Australian context, which has introduced predators, in this case mainly foxes and cats, competitors such as rabbits, and in some areas land clearing and changed fire regime. The subspecies or the population we'll be talking about today is this one in the centre of Australia, which is Central Australian subspecies of the black-footed rock wallaby, and it's currently got a listing of vulnerable, as you can see there. And there's just a couple of pictures there of a couple of the other rock wallaby species to show you the diversity that there is. So the black-footed rock wallaby is interesting for a whole lot of reasons, most of which I won't be talking about today, but it consists of 5 subspecies that all have different chromosomes. So these are kind of like the wetters of Australia. They have lots of chromosome variation within some taxa and between taxa. And so we have fusions and other sorts of rearrangements within each of these 5 subspecies, and they're also morphologically quite distinct. But today we'll just be talking about these pink ones in the middle, which is the Central Australian subspecies, Petrogale, lateralis, centralis. But it is known throughout the desert communities in Central Australia as warru, and so that's been adopted as the common name for this subspecies, and I'll be using that throughout the rest of the talk. It's also much easier to say than Petrogale, lateralis, centralis. Okay, so this is former distribution of warru through Central Australia, and then there was a massive collapse in both abundance and distribution, and so we've ended up with just scattered remnant colonies and lots of extinctions. Most of the extinctions have occurred in the southern part of the range, from within South Australia and the Southern Northern Territory, and also in parts of Western Australia. The actual populations around Alice Springs themselves in the McDonald Ranges are still reasonably healthy. And so it gives a really biased perception, because most people see that this species is still fairly common around Alice Springs and don't realise that in the other 80, 90% of its range, it's actually in a really bad way, but it seems to be doing quite well just around Alice Springs in the McDonald Ranges. So I'll be talking about these populations right up against the Northern Territory, South Australian border. And these, the Devonport Range population you see there in Central South Australia went extinct in the late 1980s, early 1990s. And what we were left with was just a couple of populations right up against the border in South Australia, Northern South Australia. And this occurs in a pretty interesting part of the world and pretty gorgeous part of the world, which is called the Anangu Pitjantjatjara Yankunytjatjara lands. Everyone refers to it as the APY lands for obvious reasons. It's a lot easier to say. Now, this is a huge area of Aboriginal owned and managed land in north of South Australia. It's basically the size of Tasmania. So it's a huge area, over 103 square kilometres. It's very sparsely populated, though. There's only about 2,300 people living in the APY lands. And it's an area of lots of rugged ranges and gullies and valleys, and some sandy country as well. So it actually supports really high biodiversity, particularly in plants and reptiles and insects, but also in a number of other species. A new species of skink was described there only last week, for example. As Aboriginal owned and managed land, it is closed to the public, so you actually need a permit to enter this area. You just can't rock up and go in there. But it is a spectacularly beautiful part of the world. So what I'll be talking about today is I've just played a very small part in a massive overall program, which is the warru recovery team. And it is involved like many people and many different players. And, I just play a small part in this overall program to manage and try and recover the populations of warru in South Australia. As I said, because all of our work is done in the APY lands, then the actual project itself is overseen and managed by a group of senior women, the warru Minma, and these are a group of senior ladies that have cultural knowledge and high standing in the community. And so everything we do has to be run past the MINMA, and they've been incredibly supportive and incredibly valuable source of both support and knowledge. The on-ground management itself is done by a group of warru rangers, which are local anangu from the local communities. And these guys are employed under the federal government Caring for Country program. So they're paid to help manage the warru and other threatened species in this area. And being a very remote area and limited economic opportunities, being a warru ranger is actually a highly coveted position in these communities because it's a job and a regular income, and you're out on country doing the sorts of things that these people really love and appreciate. So the recovery team is made up of representatives of these two groups, but also all the usual people you'd expect on a recovery team of scientists and government, wildlife managers and the zoos and all those sorts of people. So it's a really diverse group of people who've done an awful lot of work over the years. And as I said, I'm not taking credit for any of their work. I'm just talking about it because it's pretty interesting and I've had a small part in it. So the brief history of the warru, the wallaby in this part of Australia, is it used to be super abundant right through all these rocky ranges. So in the 1930s, anyone who went into this country commented about how many warru there were around. By the 1990s, it was evident there was really massive decline. There were lots of populations going extinct and disappearing right through the whole area. So that by 2006, they were only known from 3 sites. And one was this site called Calco, which is right up in the corner there. You can see it adjacent to the West Australian border. And then the other 2 sites were quite close together in that blow up. You can see they're called Alauka and Newell. You'll see them there in that little blow up. And so they were the only 3 sites where warru were left in South Australia. All of these populations were quite small. They were probably some sites only had probably 10 individuals, some maybe 20. And so we knew we'd have our work cut out for us to try and bring these back. But, first of all, we had to ask the question about whether we should bring them back because they still occurred elsewhere. Do we put in the resources to manage these populations and try and bring them back? Or do we just let them quietly go extinct and concentrate on other things? I guess in consultation with the local traditional owners and government officials, it soon became apparent that these animals, there was great reluctance to let these animals go extinct. And I guess that part of that fits into the overall picture of the appalling extinction record that Australia has seen for mammals over the last 200 years, where we have at least 40 species we know have gone globally extinct. Most of those have occurred in the arid zone, including in this area. And there's been many additional local extinctions of things that are no longer found in the arid zone, but haven't gone globally extinct. Now, most of these have been small to medium-sized mammals, so in this sort of range, greater than 35 grams and less than 5.5 kilograms. And the rock wallaby was actually the only medium-sized mammal left in the arid zone in this part of the world. And all these other species you see here had gone extinct in this part of the world. And many of them are, both culturally and ecologically really important species. And so the local traditional owners were really reluctant to have another species go extinct on their watch. And so there was tremendous desire from them and from the South Australian government wildlife people to not let these populations go extinct. So yeah, even things like the brush-tailed possum, which I know is not greatly loved in your part of the world, was a really important, culturally significant animal in this part of the world, which disappeared in the 1960s and 70s. So in 2007, the warru recovery team was formed and on-ground management started at the sites where the warru survived. And that involved managing pests and fire and some weeds as well, so mainly cats and foxes and other management around fire. Some of those populations actually responded really well to that on-ground management and started to increase almost straight away. Others, it's been much more of a struggle. As part of, because the numbers were so low, then a captive breeding option was being considered. But because numbers were low, there was great reluctance to remove any adult animals from these small remaining populations. And so the very clever folks at Zoos Adelaide and Adelaide Uni came up with a strategy of actually removing pouch young from wild females. So taking the young out of the pouch, transferring it down to Monado Zoo and cross-fostering it into yellow-footed rock wallabies who would raise that young. And then the female warru would have, because they're continuous breeders, they would produce another young a month later. So this was a way of harvesting, if you like, individuals from these populations without taking out adults. And after a lot of discussion and consultation, this was approved by the MINMA, and seen that was really the only option we had of being able to set up a captive breeding program. But they were very, very keen on the fact that if animals went away, they had to come back. And so we were happy with that arrangement. But there was a lot of cultural stuff to get through to get to that point where everyone was on board. So those very clever guys did that. They took 28 pouch young over a 4-year period. While that was going on, a predator-proof exclosure was built in the APY lands near some of the available sites, already enclosed a couple of rocky outcrops, as you can see there. And the purpose of that was to exclude cats and foxes from an area where we could put these captive bred animals back in. And so that was built. The pests and rabbits were removed from that. So foxes, cats, and rabbits were removed. We actually left some of the big goannas in there, even though they do eat rock wallabies, so that there was at least some sort of predator in there to keep the wallabies on their toes a bit. And that enclosure, the locals called it a pinchy. And so that's the name I'll be using from now on to refer to it. So when I say pinchy, I mean this 100-hectare exclosure built up in the APY lands. So in 2011, 16 captive bred warru came back and were released into the pinchy. So they came back from Adelaide Zoo. And these were from the Newell Alauca population. Down in the zoo, they'd actually kept animals from Calca separate from the animals from Newell and Alauca. The Newell Alauca animals being only being about 20 k's apart, everyone was happy for them to be mixed because they were actually still in demographic contact with each other. But the Calca population, because it was 300 kilometres away, initially was kept separate. But then a few years later, 2015, after you'll see from the data, some genetic issues started arising in the pinchy, then we combined all of these populations together and managed them as a single population. So in 2015, 5 animals from Calca stock were put into the pinchy to hopefully boost the genetics, and we'll have a look at that a bit later. Animals did really well in that enclosure, and so then we had all these animals that we had to do something with and release back out, which was the ultimate aim, to try and bring them back across the lands. And so in 2017, a mixture of animals, captive animals from the pinchy and wild animals from the Newell site were reintroduced to a new site at Aotearoa. This was the site where they had last gone extinct in 2005 in the lands. And so we put them back there under pest management. And then in 2018, we added some additional animals to add new genetics from the Northern Territory into that. And then over these last 5 or so years, as the numbers have built up again in the pinchy, we've had lots of animals to deal with. So we've supplemented Aotearoa again. We've also supplemented the wild population of Calca, and now 2023, we've done another reintroduction to another site a lot further south where they became extinct, probably in the 1950s. So it's very sorry for all that background, but it'll help you understand the actual genomics data when we get to them. So some very small beginnings, lots of stuff has been happening. The reason we could harvest from the New Well colony wild individuals was that it has done really well under feral pest management. And so it now numbers well over 100 individuals, so we're able to harvest from it as well. The other sites, less so. So moving on to the methodology, all of these populations are actively managed by the recovery team and the warru rangers. Populations are trapped and individuals are tagged and microchipped so we can keep track of individuals. And so there's regular field work monitoring the populations, and every time a new animal is captured, it is genetically sampled at the same time that it is ear tagged. And so we have a really good collection of genetic samples from all of these animals. So coming to the actual questions we're hoping to address, really they're a fairly standard set of population genomics questions, but people don't always go back and assess how successful they have been in these things, and often just assume things have worked. So it's really valuable to be able to go back and have a look at the success or otherwise of your different management options. So first of all, we're looking to establish baseline levels of diversity in the wild populations to see what we were actually dealing with. And so how diverse were they, how differentiated were they? Look at the diversity that have been captured in the zoo populations and then subsequently in the exclosure, the pinchy population. And then for these reintroduced populations and supplemented populations, how successful had we been in terms of integrating new genetics, both in the pinchy and in the reintroductions. And of course, use these data and the ongoing monitoring to inform ongoing management of this species in South Australia. So as part of the Oz Mammals Genomics, all the genomic data was developed as part of the Oz Mammals Genomics, or OMG, initiative. And I'll just give a little tiny bit of background on that before we progress. So Bioplatforms Australia, which is really the Australian equivalent of your Genomics Aotearoa, funded all this and set up an initiative called the Oz Mammals Genomics Initiative, and it really had three major objectives. Firstly, to produce additional high-quality marsupial genomes, to produce a comprehensive phylogeny of Australasian mammals, have everything a framework for future analyses, but also generate a lot of population genomic data for threatened Australian mammals. And so through this program, there's probably about 20 different species have had population genomic data generated for them, and one of those is these populations of warru. But as part of OMG, we also have generated rock wallaby genomes. We've got whole genome resequencing from a whole lot of populations. We've got DD-RAD data that I'll talk about today, and also exon caption data. So really a big effort to generate a lot of data from this very interesting group of marsupials. And so as I said, I'm only talking about the DD-RAD data today. There's a lot more data we've got out there. So from the 222 animals that we sampled from both wild captive enclosures and reintroduced, we did DD-RAD sequencing and then fairly standard processing of that data and filtering to then come up with about 2,300 SNPs, which was a bit lower than I was anticipating, but yeah, we can talk a little bit of that later if anyone's interested. And then developed fairly standard genetic diversity matrix in standard programs like that. Okay, so finally, after all that background and summary, some actual data. The first panel there, you can see with just looking at differentiation amongst the wild populations of warru based on these SNPs. You can see that the populations in South Australia really fit into two clusters, which is hardly surprising. They're about 300 kilometres apart with no recent gene flow, and so they're quite differentiated there on the y-axis. And the populations in Northern Territory, which we also included there, are again quite different on both the axis 1 and axis 2. We just take that little cluster of populations in the centre of South Australia and look at the PCOA of those. You can see, again, there's some level of differentiation between those closely, geographically close populations, but they're not completely homogeneous. There is some differentiation there, although we know animals do move between that Newell site and that Kunkamunka site. We know animals move between those two sites, but they don't that often exchange genes because they're still differentiated. Then the table at the bottom just gives a basic summary of the stats in terms of allelic richness, corrected for sample size, and then observed heterozygosities. So diversity is highest in the telegraph station population, which is in the Northern Territory. That's a population that hasn't been through a bottleneck. It's stayed relatively abundant over the last 200 years. And so it's got really quite good levels of allelic richness and observed heterozygosity, and then all the rest of the populations in the APY lands, which we know have been bottlenecked because they crashed, have lower levels of diversity, particularly that Khalkha population, which is probably numerically the smallest and has not recovered very well under management. It's got lower levels of diversity. So that's really good to know what we're working with. If we just look at the zoo population now, as I said, initially it was managed as two separate populations, and then it was mixed in 2015 into a combined single population. And you can see that initially the zoo population had better diversity than some of the wild populations, but less than some of the others. But once we combined them all together into one population, then you had a significant boost to diversity within that population. So the Monado post-Khalkha population, you can see down there in the bottom right, has a much higher observed heterozygosity than the pre-mixing. And so we're happy that that decision to mix them had resulted in a significant boost to diversity in the captive population. And you get similar results for allelic richness. When the animals are introduced into the Pinchi, if you just look at so that was founders just from Newell and Lauka. You look at their offspring from the 2012 to 2015 period. You can see that as the animals from Lauka and Newell bred with each other, they started in this second panel in the centre there. You can see that the Pinchi recruits from that period started to develop their own genetic characteristics as they merged between the Lauka and Newell genotypes. Interestingly, though, as often happens when you do a reintroduction or set up a captive population, there was a net loss of diversity from the founders that were put into the Pinchi, and then the actual animals that produce the actual population that then established from those animals. So there was quite a significant drop in diversity because not all the animals put in bred. And so you actually ended up losing quite a bit of diversity in the establishment of that population, as you can see there in that bottom right panel. And again, this is not uncommon, but people don't often go back and have a look at what happened during that whole process. And so confronted with this, we knew we had to supplement the Pinchi population again to try and get that diversity back up. And so then in 2015, we put in the Khalkha derived animals from the other population. And you can see here that initially they're all quite separated. But then in the second PCOA, you'll see that as animals start breeding with each other, then in the new set of offspring from the 2016 to 19 period, you're getting much better mixing. Some individuals are drifting over towards that Khalkha population as individuals from Khalkha breed with the other individuals, and you're getting a boost to the genetic diversity in that population. And so although in 2015 it was a little bit controversial to be mixing populations, we're all kind of over it now and people are much more relaxed about mixing populations. In 2015 it was a bit of a more controversial move. One, I have to say that the TOs were incredibly supportive of it, encouraged, but there's still some reluctance in some quarters about mixing at that point. Now, as I said, I think we're all a bit in wildlife management circles a bit more relaxed about it, but you can see that it certainly had an excellent effect in terms of boosting the diversity of the pinchy animals. And then as the numbers in the pinchy grew, we had to take them out so that we wouldn't have overabundance in the pinchy. And so in 2017, we began a series of reintroductions and supplementations, which took over the next few years, have taken about 150 animals out of that pinchy without affecting the demography or the demographics of what's in there. So they produce, when not being predated regularly, they really can increase in numbers quite rapidly. So we picked really diverse founders to go into this other site, this Aotearoa site. And they had been derived from Northern Territory and from the pinchy and from the wild side at Newell. And again, you can see there in that second panel that the recruits born from the Aotearoa founders, because the founders were so diverse, you actually had a real boost in the diversity of the recruits. So instead of losing diversity, you actually gained it because we had really diverse founders being put into this site, which is really good to see. And we're very pleased that we're able to see the results of mixing there, both in the observed heterozygosities and in where the recruits are placed. Okay, so I didn't want to go on for too long about all that because I want to leave some time for discussions. So that's where we are at the moment. As I said, there's been additional reintroductions since then and supplementations. We have all those samples which are in the process of analysing to see whether we've been similarly successful. But in terms of, I guess, the take-home messages from all that is that what we're able to show from using that reduced representation genomic data was that the surviving populations in the APY lands did have reduced diversity, that all the populations were differentiated from each other. Part of that is natural, but it's probably been exacerbated by population extinction and the more recent loss of gene flow between them. The Kalka population in particular, that one right over in the corner near South Australia, was very low in numbers and also very low in genetic diversity, and so we recommended supplementation of that population, which has now occurred from animals out of the pinchy. And we're able to show that our strategy of mixing populations, both in captivity and in the exposure, have successfully increased the diversity, genetic diversity of those individuals in both the captive situation and in the pinchy, and that even introducing a small number of new individuals, even into a larger population, that they can successfully integrate and contribute their genes and are not being outcompeted, demographically or socially. It's also clear that, most of the gains we've made in genetic diversity have been fairly modest, and so we need to keep going with additional supplementations. And as currently, most of these populations are still closed and are not going to get natural gene flow, then there's going to be an ongoing need for that supplementation. Certainly the strategy of mixing populations in reintroductions from a diversity of backgrounds has resulted in really diverse recruits being born in that population, that you're not getting a loss of diversity, but you're actually enhancing the diversity and setting, hopefully, those populations up for a really good future. The other, I guess, two takeaways is one that, wildlife conservation is you spend a lot more time talking to people than you do dealing with animals or data. And that so the success of these things is often totally dependent on, good communication with people and spending a lot of time talking to people so that you can then actually do the wildlife management. And in this particular context, we're very privileged to work in this cross-cultural environment and have learned that, modern science and traditional knowledge is a really powerful combination, and that you can achieve, great things if you're willing to sit down and talk to each other and listen to each other and exchange knowledge and views, and then develop strategies moving forward. Lastly, I just need to really thank my two collaborators here, Dr. Sally Potter from Macquarie Uni and Dr. Rebecca West from University of New South Wales, who did most of the legwork on this genetic data analysis. The warru recovery team, of course, Bioplatforms Australia for funding, and the Oz Mammals Genomics team. And then all these people at the bottom are the ones who actually help run and manage these programs in South Australia. And as I said, the genomics component of this is really just a very small part of whole groups, massive numbers of groups of people all working together to try and bring back the warru in this part of the world. And so far, so good. We seem to be doing well. And I think it's a really nice success story of working cross-culturally and with modern technology and traditional knowledge to actually bring a species back that could have so easily gone extinct. So, yeah, with that, saying thank you very much and happy to take questions.